everybody, welcome to the Inspiring Minds coaching. And now it's time to go one step ahead in the microbiology section. As in my last video, I told you about the classification of the microbes according to the temperature and according to the oxygen availability. Aerobes, anaerobes, obligative, and capnophils. And later I will give you the more descriptions when I will talk about selective microbes. Now, today's topic we have the bacterial structure. What happens when we go through the staining process or somehow any microbial investigation? So there are some specific things which present in the bacteria already, and they show their effect with the proper dye or one kinds of any kinds of you can say that like activity or experiment which you do. So there it's very important for you, I mean for everyone who desire to learn the microbiology, that what are those structures which specifically present in the microbes. Not only microbes, even when you study other pathological process also they are present. Okay, so let's begin. Bacterial structure and its component by the way I will discuss with you later, but now in this slide I am going to just deal with the bacterial structure. Most probably every time when you see the question and also when you understand some kinds of questions, so it becomes very difficult because we get confused that which kind of things are there and how we can discriminate with them. Here are some simple tricks which is very important to understand from the very beginning. That this time taking you to my first video, and there I introduce you about the introduction of the microbes. Again revising it, that guys, we are living on the earth, and earth is composed of living and non-living materials. Livings have the according to Whitaker classification, the 5 kingdom of classification. And similarly, before that, Aristotle, the father of biology, he gave the hypothesis of the living organisms, and there was a lots of evolutionary development. Okay, so living have basically two types of categories: micro level and macro level. Microbiology not only deal with the bacteria, but all those organisms which we are unable to see with our naked eyes. Okay, so here the microorganisms and macroorganisms. In the macro, like we have animal, plant body, or some kinds of we can say fungi, lots of big plant like a big body. So here moving toward the moving to the bacteria, as just to refresh it in your mind, that Monera. Before that it was acellular life, and there, if you remember, that was a very famous term which was used, amoeba and paramecium. So similarly here we have the first one where we define the cell. Okay, a cell body. That's why here we talk about the 5 kingdom of classification where the cell body existence, and the first one, Monera, where we say the prokaryotes. Prokaryotes means false nucleus but nuclear material present. Remember my second video where I told you about the description before staining process. And even in the first video also I told you the nucleoid which present in Monera or in the bacteria. So it is more deeply what exactly present in the bacteria. Guys, what happen in living body? If any living body, whenever it goes to the classification or in the evolutionary process, day by day or year by year, or you can say in the evolutionary development, there are some structural modification and also physiological modification which you already study in the adaptation process of ecology. Okay, so here that Monera is prokaryotes and bacteria. Obviously if there is some kinds of, because before that was acellular structure, now it is cellular structure and there is composed of cells. So there is we are going to talk about the small body microbe, that is bacteria. Okay, any body contain, if I talk about anyone like us, or somehow any animal or anything you can see living object, so they have one core part, body, and the additional one, there are appendages, which is very important, and somehow in later modification they have head. Okay, in the animal's case. Similarly, if I consider myself as a body, so my trunk part will be considered as trunk, the body, and the appendages will be my hands and leg and head. Okay, remember the diagram of the bacteriophage. But here is something I am giving you the example how to just to see the similarity. The protozoan causing malaria disease is very famous in many licensing examinations. So that's why you should remember that protozoa is not any bacteria or protista or fungus. It is a protozoa. In another kingdom we study, that is not a kingdom, but it is a cellular structure. Okay, so here in the bacteria, what are those four subdivisions of the bacterial structure? The first one is the body, and second one appendages, third one special structure, and the fourth one cell envelope. On accordance with the pathogenesis and all these things in our modern words, that you can say that why this problem is caused by the bacteria. Basically they are derived from the cell envelope. Okay, you can say that there everything is formed, but this one is like gate from where a lots of any kinds of toxicity or any kinds of harmful substance released from this gate. Okay, this entry. So the body part of the bacteria contains cytoplasm and nucleoids. Sorry, the cytoplasm and nucleoids is contained here. It's not nucleus because it is prokaryotes. Okay, cytoplasm is similar to the eukaryotic cell, and nucleoid, as in my first video I told you about the genetic material in the bacteria. Okay, going ahead, we found appendages. Appendages is basically two types which present in the bacteria. The first one flagellum, and the second one pili and fimbria. Don't worry, specific function and their components I will discuss in the next video, but here in this video I'm just going to give you this idea, and I will tell you that from where question asked and from where you should while we read the question, some especially in the FMG examinations and also in USMLE examinations, what happened there, they give the specific structures directly so that you can understand. If they are talking about the cytoplasm and nucleoid, definitely they are going to deal with the genetic answers. Okay, molecular level diagnosis or molecular level pathogenesis. But if they are talking about pili, fimbria or exotoxins, so it means they are talking about external features of the bacteria. So similarly it will be cellular level dysfunction or pathological changes, right? So here after the body appendages, and there is a special structure of bacteria that is known as spore. Remember in my last video there I told you about the sporulation process. I think it is very famous in the bacteriology, even not only systematic bacteriology but the basic bacteriology, that sporulation is produced by the process which is known as sporulation. And why they produce? What happened? See guys, we all have a proper demand of environment, proper temperature, proper pH, humidity, and air velocity. All these things, micro elements in the ecology you will study, hygiene and ecology, that we all need to survive. But what happened that when something is not under our our physiological demand, so what we do? We just define ourselves as a physiological adaptation. Like breathing rate increase when we go to the high altitude, so that is very famous for you if I talk about the RBC's structure and their hemoglobin binding capacity when you talk about the pathomorphological changes. But here in simple way, when there is a summer condition we sweat, okay, when it is cold condition we shiver. So all these things are our physiological adaptation. But what happened to the bacteria? Somehow microorganisms are the very basic structures, so they don't have that kind of adaptation because they don't have that much neurons and that much nervous system elaboration. Neither they have endocrine system. That's why they have some kinds of cellular modification. It's not just a modification, but they keep them survive for years to years, thousands of years. Okay, right now lots of living organisms already present on the earth, which they are still here from the last 1,000 and more than 1,000 years because of their cellular modification. They have a small body. So spore produced just to survive in that extremely opposite or against circumstances. Like there is a lot high pH, okay, and you can say that other high pH and temperature high, and also some kinds of problem happen when we talk about the spore function, because spore from exam point of view, very important, just it's for you to remember that it only and only found in gram positive bacteria. Whenever you solve the question and there is a term used spore, make sure that that will be definitely gram positive bacteria, right? So now the third one is goes to the cell envelope. Cell envelope is most important, and also most of the question and staining process, lab technology and growth medium functions, all these things are defined only behalf of the cell envelope structure. What are the cell envelope structure? Before starting the cell envelope, I want to make it clear that what does it mean envelope? You have something, some kinds of cover and covering yourself completely, okay? So it means that you enveloped yourself. So make sure that envelope, like similar envelope, we just when we send the letters, what we have an envelope where we keep that letter, and it should be properly transported to that person so that no one can interfere, no one can there will be no external damage, and also protection will be there, okay? That's why envelope is important. So what about cell envelope? What does it mean? Cell envelope means simply it is going to envelop the cell. So you can just understand what are those structures and modifications of the bacteria which help them to preserve, right? So like the first one was the spore, but spore is not to preserve whole body, it preserves the core material, okay? So here the cell envelope is very important, that the first one is capsule. See here I have mentioned with the blue color capsule. Capsule present in both gram positive and gram negative bacteria. Capsule is the outermost layer. Just you can remember, you can imagine like having any child or any person is inside the balloon and going to the water, and so there is balloon like a structure outside. Remember, same capsule like that. It's not a pharmacological capsule, that is another thing. Somehow that is also kind of envelope there present, but you can just go through them that they are similar but not same, okay? Their property are different because there are absolutely that pharmacological drug that are the non-living objects, they show their effect toward the living object and living subject, but they are not a living thing, okay? But here capsule is a living, it present on the living things. So here the capsule structure I will tell in later that their component and their function. Capsule present, and the second one, slime layer. Guys, in the growth media I told you about various selective and basic classification of the growth culture. So there I told you that some of them form the slime layer, some of them form the rough layer. Actually these are their characteristic property to identify the specific type of the bacteria, right? Not only the bacteria, even if you remember that cellular level eukaryotic cells, what are there are rough endoplasmic reticulum, smooth endoplasmic reticulum. Smooth endoplasmic reticulum, somehow we talk about the smooth shape, right? So here the slime layer, and the third one is a cell wall. Cell wall is simply that structure which is present like a wall to protect the cell. We live inside the room and we have the wall. Similarly, there is a cell wall, right? So here cell wall is basically their composition of the cell wall, peptidoglycan I will discuss, but you should keep in mind like basic concept that cell wall present. And after cell wall there is a periplasm. Periplasm, what does it mean, periplasm? Plasm is somehow related to the cytoplasm, and peri means outermost. Remember that when we study this basic mathematics and we talk about perimeter, or leave that mathematics, if you go to the biotechnological section, there was the centrifugation, or in the physics you have centripetal and centrifugal, okay? There also when we talk about the perimeter means that outermost layer, the outermost content and outermost their observation is referred to the periplasmic observation, right? Plasm is similarly related to the cytoplasm. Cell, plasm, cell fluid. Periplasm, it means fluid outer boundary, means that boundary till till that place the cytoplasm present. That is periplasm. Okay, periplasm, and after then there is the cytoplasm membrane. Cytoplasmic membrane, why? Because it is somehow almost 70% is composed of the cytoplasm, and membrane because it from the very thin layer of the cytoplasmic boundary. What happened that when we study about any bacterial structure, these all things get mixed up. 3 times USMLE have been asked this question, their sequence. And it is very famous question for the NEET PG also. So similarly, just their sequence is very important. But if you use your general or basic knowledge, then definitely you will be able to solve these questions. Because just keep in mind, like we have, bacteria have the cell structure. In cell structure, the core one nucleoid. Then there is cytoplasm. Cytoplasmic organelles will be there. No, membrane bound cytoplasmic organelle is absent, right? So here that cytoplasm and the outermost will be the cytoplasmic membrane after cytoplasm just, and then there will be periplasm. If you just match the word cytoplasm, periplasm, plasm match, then there will be one step ahead, the cell wall, cell this match, and then there will be the slime layer, outermost slime layer. Okay, so you can just with the layer, and in the last when everything is ready, like some child is ready, then we put the cap. So just remember that small story that when you have, bacteria have their nucleoid structure and their cytoplasm, they have a great responsibility to to survive and live the normal life. So they just have their nuclear structure, they have their body or core part, and then they have the cytoplasmic membrane, the outermost, then periplasmic membrane, then they have the slime layer and cell wall, then they have the slime layer, and then the last one they have the capsule. Capsule is not every time, but mostly capsule present in both bacteria, gram positive and gram negative. Don't be confused that gram positive bacteria have the thick layer. That doesn't mean that they don't have the capsule. They have the capsule. Okay, that's why the pathogenic property of the gram positive bacteria is more prominent as comparison to the gram negative bacteria, right? So here the last one is here the cytoplasmic membrane. After cytoplasmic membrane, cytoplasm will be there, and then spherical bound there will be nucleoid and extranuclear material of the bacteria, that is plasmid. Okay, remember that I told you that most of the bacteria just reproduce with the binary fusion, but I told you whenever there is a question and one sentence is used, each and every bacteria reproduce through the sexual reproduction, or you can say the binary fusion. That is completely wrong. Mostly, but not all, right? So here I am telling you the bacterial cell envelope structure, because the later whole bacteriology or microbiology is going to depend on the cell envelope and its structure, function and component. But when I talk about the molecular level genetics or molecular level of clinical biochemistry, every time we are going to discuss nuclear material and the body part, this one. Okay, so here in the structure I have mentioned my left side gram positive bacteria cell envelope and my right side that is gram negative bacteria. You can see in the video that same will be. So you can see guys, there are few structures which is common. So I have mentioned, and I am going to draw a simple dotted line so that you can just imagine, you can remember that these all things are common. Okay, common. And what is differential thing and what is very important to remember, and in the questions many times it has been asked because of the special things which present. Here this and here this. So just quickly see the last the cell envelope and we will finish this video. That the flagellum, okay, I remember it with the F. That same thing is here, flagella. Flagella is present in both of the bacteria, gram positive and gram negative, mostly bacteria but not every bacteria. Villi, similarly, they present in both. Here I have represent P and fimbriae, Fi small. Then cap present in both bacteria, that is capsule, and I have mentioned earlier that they are present in gram positive and gram negative bacteria, and they help them to protect from the phagocytosis. And also the cell wall peptidoglycan layer, that is present in both bacteria, but you can see that thickness of gram positive bacteria is more than the gram negative bacteria. That's why gram staining process it help us to differentiate between the gram positive and gram negative bacteria. Remember the first video where I told about gram stain process, 6 minute 6 step gram stain process, and all questions I gave you the final trick to solve all questions. And then the periplasmic layer, you can see here that the outer membrane, that one is a periplasmic layer, this one, okay, this one is periplasmic layer. Or to make it more clear, I will just draw with black. That is periplasmic layer. And then there is cytoplasmic membrane. Cytoplasmic membrane, phospholipid bilayer in the animal. So keep it in your mind that there is also similar structure present in bacteria. Okay. So and what is very important and very famous to the question point of view, that gram positive bacteria contain teichoic acid, lipoteichoic acid according to USMLE, and teichoic acid according to the NEET PG, FMG examinations, and also there is lipoteichoic according to the rock questions and PLAB. So keep in mind. And similarly in the gram negative bacteria, they have the special proteins which present in their cell wall, that is known as porins. They have the facilitated transport system and also help bacteria to survive and to produce the specific bacterial function. Endotoxin and porins. In the next video, I will give you one by one the explanation of all these things. But here, just remember that lipopolysaccharides or the endotoxin means internally produced, exotoxins means outside to produce. Endotoxin, here the outer membrane of the cell envelope is able to produce endotoxin. That is the gold standard marker for the gram negative bacteria. So everything is there, and just you can see in the quick revision point of view. And hope you guys like this video. Please like, share and subscribe the Inspiring Minds coaching, and be in touch with the microbiology. Just in each and every video, in the last, I give you the best and simplest thing to keep in mind and help you solve the questions. Remember the star mark points, which are the examination point of view, very important. And from in the next, here some diagrammatical point of view, and from the next video I will make it theoretical approach and explain the most questions which have been asked with them in various examinations. Thank you.